Okay, now we are going to talk about DNA replication. What is DNA replication? DNA replication happens when the cell is going to be divided, so the chromosome is going to duplicate its genome. How this process occurs? We have a region on DNA that we call it replication region, which we have a high rate of AT base pairs. Like for example, it can be like this. So the parallel strand will have T, T, A, T. The replication will be started at this region. But why? Because between this region, between the bases of this region, we have two base pairs. Two hydrogen bonds. But if it was G, C, G, G, C, G, C, C, for example, there were 3 hydrogen bonds between them, so it's harder to break them. Therefore, the cell or DNA started replication from this part. How it occurs? DNA has 2 strands, okay? We say that DNA replication is semi-conservative. What does it mean? It means that semi-conservative. It means that during the replication, we have one strand. After the replication had been done, we have one strand that is from the old DNA, and another strand is new and newly replicated or produced. So, at the first, these 2 strands should be separated from each other to make a replication fork. But how does it happen? By the help of an enzyme that its name is helicase. Helicase will break the hydrogen bonds between the 2 strands. So we'll have something like this. These are the 2 strands that were broken, and this part still the helicase is processing, it's breaking the hydrogen bonds. For example, it's the hydrogen bond. Here they were broken. I try my best to be fast in it. So here we have helicase. I just write edge. Helicase starts to break these hydrogen bonds. But when this breaking happens, what will happen to the strands? It will make a kind of torsion, like the strain will be torsioned. So maybe if it wants to continue its way, the helicase, it cannot pass anymore through the double strands to break the hydrogen bonds. Therefore, we have to have another enzyme, okay, that we call it telomerase. topoisomerase. I was mistaken. So we'll have another enzyme that we call topoisomerase that they reduce the torsion of the strands and the positive kind of we can say positive supercoils. So topoisomerase reduces the torsion and positive supercoils. Why? Like, I'll tell you now. For example, this is the helicase here that is breaking the hydrogen bonds, that is breaking them, but they will get, the strands will get mixed matched together, they will get like torsions. So topoisomerase will reduce it. Now, if we want to continue the replication, we should have a sequence that we call it RNA primer. RNA primer is made by the nucleotides, the RNA nucleotides, that it will be appeared on the starting point of the DNA polymerase. For example, I show it in, let me say, by green color. Okay. This primer, this sequence was made by an enzyme that its name is primase. Now we should have another enzyme that we call it DNA polymerase. DNA polymerase will recognize this primer, and then after that it will start the replication by adding the nucleotides. But how? Like the nucleotides, how they will be added to in which direction it will be added to the DNA. Let's talk about this. DNA molecule is antiparallel. What does it mean? It means that when it is 5' here, the other side of the molecule is 3'. When it is 5' here, the other strand will be the complementary part of it, means the 3', the other side will be 5'. So when the DNA polymerase will recognize RNA primer, then it will be attached to the original part of the replication. It will start synthesizing the new strand of DNA from 5' to 3' direction. So it can only synthesize the DNA from 5' to 3' direction. Therefore, here we will have a continuous way of like DNA synthesis for this strand from 5' to 3', that we call it leading strand. Okay. But on the other side, on the other strand, the replication also will happen, because the replication is happening simultaneously on the both strands of the DNA. But how it is possible? Because here this strand is 3', 5'. If, again, the same as before, we want to start a DNA replication from this part, it will be from 3' to 5', that it's not possible for DNA polymerase to do so. It can only add nucleotides let me only add nucleotides from 5' to 3' direction. So what will happen? In this case, we will have, as before, RNA primer that will attach to a sequence of the other strand of DNA, that it will be again 3', 5'. Then after the DNA polymerase starts to add nucleotides from 5' that were made by the RNA primer to 3'. Okay, then after again the helicase will start to continue and make the DNA much more open, so we will have, means it starts to move forward. Okay, for example, these hydrogen bonds were removed, but I don't like if it will, oh no, okay, this section is, for example, it will happen like this, in this way. Now again we have the continuous of the open strands of the DNA, that here again it is 3' and 5'. So another RNA primer will be attached to the 3' to the 5' end. Again the DNA polymerase will recognize the 5' and starts to add nucleotides from 5' end to 3' end. So now it is able to do it, but you see that it's not continuous. Again here it will be like this, 3'. Okay, it's not continuous, this is discontinuous kind of replication, and each section that the DNA polymerase made it, we call it Okazaki fragment. And we call this strand lagging strand. Okay? In the end, the RNA primers will be removed, will go away, and these discontinuous strands that were made by the DNA polymerase will be attached to each other by an enzyme that we call it, like will be attached, for example, like this, by an enzyme that we call by a DNA ligase. So this is the initiation part of the DNA